Hello friends, today let us learn about sulfur containing amino acids. Sulfur containing amino acids are methionine, cysteine. So methionine in the presence of ATP become S-adenosyl methionine. Methionine has a linkage called thioether linkage. It is stable. It has a thioether linkage. It is stable. It reacts with the ATP and releases inorganic pyrophosphate and gives you S-adenosyl methionine. It is a methyl donor. So the acceptor accepts methyl group from S-adenosyl methionine and the S-adenosyl methionine become S-adenosyl homocysteine. This S-adenosyl homocysteine releases adenosyl group to give you homocysteine. So methionine become S-adenosyl methionine by the enzyme methionine adenosyl transferase. S-adenosyl methionine become S-adenosyl homocysteine by the enzyme methyl transferase. S-adenosyl homocysteine become homocysteine by the enzyme adenosine homocysteinase. From homocysteine we get methionine by getting methyl group from one carbon pool in the presence of vitamin B12 and tetrahydrofolic acid. Also from homocysteine we synthesize cysteine. So here there is N5N10 methylene tetrahydrofolic acid become N5 methyl tetrahydrofolic acid by N5N10 methylene tetrahydrofolate reductase enzyme. This N5 methyl tetrahydrofolic acid along with vitamin B12, which is absorbed from the intestine, releases tetrahydrofolic acid and form methyl vitamin B12. This methyl vitamin B12 donates methyl group to homocysteine to generate methionine. This is how we get methionine from homocysteine. Also from this homocysteine, when it condenses with the serine, it gives you cystathionine by the enzyme cystathionine beta synthase. So this cystathionine by the enzyme cystathionase in the presence of pyridoxal phosphate gives you cysteine plus alpha ketoglutarate plus NH4. This is how we generate cysteine from methionine. So the sulfur containing amino acids are methionine, cysteine. Also we generate homocysteine. So this homocysteine condenses with the serine in the presence of pyridoxal phosphate by removing a molecule of water, gives you cystathionine by the enzyme cystathionine beta synthase. The cystathionine by cystathionase enzyme by adding a molecule of water releases homoserine and cysteine. This homoserine by deamination and decarboxylation gives you propionyl CoA. So methionine is a sulfur containing essential glucogenic amino acid. It forms succinyl CoA. It is required for the initiation of protein biosynthesis. Degradation of methionine results in the synthesis of cysteine and cysteine. So activation of methionine to S-adenosyl methionine. So in the major pathway, methionine is activated to active methionine S-adenosyl methionine. The synthesis of S-adenosyl methionine occurs by the transfer of an adenosyl group from ATP to sulfur atom of methionine. This is done by the enzyme methionine adenosyl transferase. There are 3 isoenzymes for methionine adenosyl transferase, out of which 1 and 3 are of hepatic origin. S-adenosyl methionine is the main source of methyl groups in the body. The activation of methionine is unique as the sulfur becomes a sulfonium atom by addition of 3 carbon. 3 ATPs are consumed in the formation of S-adenosyl methionine. Methyl transfer. In methionine, the thioether linkage is very stable. In S-adenosyl methionine, due to the presence of a high energy bond, the methyl group is labile and may be transferred easily to other acceptors. S-adenosyl methionine transfer methyl group to acceptor and gets itself converted to S-adenosyl homocysteine. S-adenosyl homocysteine is hydrolyzed to homocysteine and adenine, which is the higher homologue of cysteine. Methionine synthesis. Homocysteine can be converted to methionine by addition of a methyl group. This methyl group is donated from one carbon pool with the help of vitamin B12. Homocysteine condenses with the serine to form cystathionine by the enzyme pyridoxal phosphate dependent cystathionine beta synthase. Absence of this enzyme leads to homocystinuria. In the next step, cystathionine is hydrolyzed by cystathionase enzyme to form cysteine and homoserine. Net result is the sulfidyl group from methionine is transferred to serine to form cysteine. This is called transsulfuration reaction. Final oxidation. Homoserine is deaminated and then decarboxylated to propionyl CoA. This finally enters into the TCA cycle, tricarboxylic acid cycle, as succinyl CoA, which is converted to glucose through gluconeogenesis. So, methionine in transmethylation reactions. Many compounds become functionally active after methylation. Protein methylation helps to control protein turnover and protects from immediate degradations. These reactions are called methyl transfer reactions, and these are carried out with the help of S-adenosyl methionine. Methyl groups are originally derived from the one carbon pool. The methyl tetrahydrofolic acid can transfer the methyl group to homocysteine. Vitamin B12 is the coenzyme for the reaction. This would account for the deficiency of folic acid associated with vitamin B12 deficiency, which is called folate trap. S-adenosyl methionine is the methyl donor for all the transmethylation reactions. These are some of the transmethylation reactions. So, methyl group acceptor guanidoacetate, methylated product is creatine. Norepinephrine, epinephrine, metanephrine, ethanolamine, choline, nicotinamide, N-methyl nicotinamide, acetyl serotonin, melatonin, phosphatidyl ethanolamine, phosphatidyl choline, serine, choline, carnosine, anserine, tRNA bases, methylated tRNA bases, lysine, methyl lysine. So, these are the some of the transmethylation reactions. From cysteine, we synthesize glutathione. Glutathione is also called gamma glutamyl cystinyl glycine. It is abbreviated as GSH to indicate the reactive sulfidyl group. Glutathione was isolated in 1921 by Sir Frederick Hopkins. Glutamate plus cysteine gives you gamma glutamyl cysteine. Glutamyl cysteine along with the glycine we form glutathione. Both these steps needs hydrolysis of ATP. This is how we generate glutathione from cysteine. So, summary of methionine metabolism. Methionine becomes S-adenosyl methionine in the presence of ATP. This S-adenosyl methionine is a methyl donor. It undergoes transmethylation reaction. So after donating methyl group, S-adenosyl methionine become S-adenosyl homocysteine. So this S-adenosyl homocysteine, after releasing adenosine group, it become homocysteine. Homocysteine condenses with the serine to release homoserine and cysteine. Cysteine by degradation gives you pyruvate, coenzyme A, taurine, glutathione. Pyruvate enter gluconeogenesis pathway to generate glucose. So when there is a deficiency of enzyme cystathionine beta synthase, there is a condition called homocystinuria. So it causes elevated plasma levels of methionine and homocysteine. There is increased excretion of methionine and homocysteine in urine. So plasma cysteine is markedly reduced. General symptoms are mental retardation, Charlie Chaplin gait, skeletal deformities are also seen. In eyes, there is ectopia lentis, which means subluxation of lens, myopia, glaucoma also observed. Homocysteine causes activation of Hageman's factor. This leads to increased platelet adhesions and life-threatening intravascular thrombosis. Cyanide nitroprusside test will be positive in urine in these conditions. Urinary excretion of homocysteine is more than 300 mg per 24 hours. Plasma homocysteine and methionine levels are increased. So how do you treat? A diet low in methionine and rich in cysteine. This will decrease the level of homocysteine. Homocysteine, how it is related to heart attack. An increase of 5 micromole per liter of homocysteine in serum elevates the risk of coronary artery disease by as much as cholesterol increase of 20 mg per deciliter. Homocysteine interacts with lysyl residues of collagen, interfering with collagen cross-linking. Homocysteine interacts with lysyl aldehyde groups on collagen and binds to fibrillin, producing endothelial dysfunction. Many patients with homocysteinemia also have morphinoid features, since the protein fibrillin is defective. It forms homocysteine thiolactone, a highly reactive free radical which thiolates LDL particles. These particles tend to aggregate and are endocytosed by macrophages and increase the tendency for atherogenesis. Providing adequate quantity of pyridoxine, vitamin B12 and folic acid will keep homocysteine in blood at normal levels. Maternal hyperhomocysteinemia is known to increase the chances of neural tube defects in fetus. So high doses of folic acids are advised in pregnancy. So this is a summary of methionine metabolism. So methionine along with adenosine triphosphate by releasing pyrophosphate or inorganic phosphate become S-adenosyl methionine. This S-adenosyl methionine is a methyl donor which is accepted by the acceptor. So this S-adenosyl methionine by methyl transferase reaction enzyme become S-adenosyl homocysteine. This S-adenosyl homocysteine, after releasing the adenosine group, it become homocysteine. Homocysteine, from homocysteine by homocysteine cystathionine beta synthase, you synthesize cystathionine. By cystathionase enzyme, in the presence of pyridoxal phosphate, you release cysteine. Also, this homocysteine by methionine synthase, you get methionine from homocysteine in the presence of vitamin B12 and tetrahydrofolic acid, which donate methyl group from the 1 carbon pool. So this vitamin B12 become methyl vitamin B12 So after donating methyl group to this homocysteine, the methionine is formed. This is about the methionine metabolism. Thank you.